Bright Duty. Every student matters.
Hello students. Today in this video, we'll study about the next chapter of biology, that is heredity and evolution. Now, have you ever wondered why humans give birth to human child? Why a dog can give birth to only the puppies? And even if you sow the seed of a maize plant, it will produce a maize plant itself. Why it is so? How come the cell know which characteristics to develop? So that is what we'll try to understand in this chapter of heredity and evolution. That branch of science which deals with the study of heredity is known as genetics. And heredity is transmission of characteristics or genetic information from one generation to the other, from parents to offsprings, is known as heredity. And the mechanism by which the information is passed on from generation to generation is known as inheritance. Now, many scientists, they tried to study the mechanism of inheritance, but it was Gregor Johann Mendel who was able to study in detail about the laws and the mechanism of inheritance, and he could produce it to us. Now, let me first tell you, the characteristics or the information, they are stored in each and every cell. As you know, all the cells have nucleus, and nucleus contains thread-like structures which are called chromosomes. These chromosomes are made up of DNA. The unit of DNA are the genes. Now, what happens? The information is stored in the genes. When the parents produce the offsprings, the information is passed on through these genes. There are thousands of genes in the chromosomes, and each gene is responsible for our characteristics. For example, your height, color of your skin, texture or color of your hair, color of your eyes, all these characteristics are determined by the genes which you inherit from your parents. This is all about inheritance. So Gregor Johann Mendel, he performed various experiments on pea plant. The pea plant, the garden pea. He used to stay in a monastery where he used to grow the pea plants. He also had knowledge of mathematics and statistics. So he applied his science and maths knowledge to analyze the results of these experiments. That is why he was successful and able to tell others how the inheritance is possible, how the information is passed on to the next generation. Before we move further, let me tell you, he chose pea plant. The scientific name is Pisum sativum. Why he chose pea plant? Firstly, pea plants are very easy to grow. Secondly, the pea plant, the flowers of the pea plant, they allow self-pollination. However, cross-pollination is also possible. So different breeds could be bred to produce new generation of pea plants. Thirdly, each plant gives rise to number of seeds. But the most important reason is the pea plants, they show contrasting characters, few contrasting characters. So it was easy to compare them. For example, some pea plants are tall while some are dwarf. Then some bear the violet color flower, the others have white color flowers. The seeds of the pea plant are either round or wrinkled. The color of the seeds is either the green or it is yellow. So thus such contrasting characters, it is easy to compare and study them. That is why Mendel chose pea plant for his experiments. Now, the first experiment which he performed was Monohybrid Cross. In Monohybrid Cross, there is a cross breeding between two plants considering only one pair of contrasting characters. Okay, so let us consider this monohybrid cross between a tall plant and a dwarf plant. Now, each character is represented by a pair of genes. So, a tall plant is represented as a pair of capital T. This is a pair of genes representing a tall plant. On the other side, he took another plant which was dwarf. For dwarf, we'll take a pair of genes which is represented by small letter t. Alright, so there is a cross breeding between tall and dwarf. As you know, what happens during gamete formation? In sexual reproduction, one male gamete, one female gamete fuse together to form a new cell, which is called zygote. Now, during gamete formation, what happens? The chromosomes, they separate out. So, gametes contain half the number of chromosomes. For instance, human beings, each cell contains 23 pairs of chromosomes, that is 46 total chromosomes. But the gametes will have only 23 chromosomes, half the number of chromosomes. So half from the male, half from the female, these chromosomes combine together to form a new individual. So in this case, when gametes are formed, each gamete will have capital T, alright? And in this dwarf plant will have the small t gametes. These are the gametes. Now during crossbreeding, what will you expect? When pollen grains of tall plant and let's say ovum of a dwarf plant, they are fertilized and a new plant is formed. You might expect that a medium-sized plant will be formed, but no. What Mendel observed was all four were tall plants. Alright, so when this gamete fuses with the other gamete, it will be the new zygote will have capital and small t. If this fuses with the other gamete, again capital and small. But if there is a chance pairing of this gamete with this, it will again have capital and small. And the fourth case also, it will have one capital, one small. This is called F1 progeny or the first generation. In the first generation, what was observed? That all the plants were tall. So that means although they had a gene from the dwarf, but this dwarfness could not express itself. So such genes which cannot express themselves in the presence of another gene are called recessive genes. And the other gene which can express itself even in the presence of recessive are called dominant gene. So that is why we have used capital T for a dominant gene tall and small letter t for the recessive gene dwarf. So it was observed that all the plants grown in the first generation all were tall, although it was expected that it might lead to a medium-sized plant. So all plants were tall. So that is why tallness is considered as a dominant gene, dwarfness is considered as a recessive gene in pea plants. Now, what Mendel did was he performed self-pollination with these plants. So when a gametes will be formed from this, what will happen? Half the gametes will have capital T, the other half will have small t. So each plant will lead to two types of gametes. Capital T and small t. Half the gametes will have capital T, other half will have small t. During self-pollination, what will happen? If this gamete will fuse with this one, it will give rise to capital T, capital T. But if this fuses with the other one, it will have one capital, one small. Now, let us consider this gamete fuses with this capital T, small t. But in fourth case, notice this small and this small, they'll fuse together to form two small t's. Right? So in this case, this will be a tall plant. Why? Because it has both the genes of tallness. This plant will also be tall. Why? Because it has one dominant gene of tallness, one recessive gene of dwarfness. So recessive will not be able to express itself. So, but dominant, because of the presence of one dominant gene, this plant will also be tall. Next, this plant again will be tall because of presence of one dominant gene. But in this case, notice, both the genes are dwarf, that is small t. So this plant will be dwarf. So in this F2 generation, what was found? That there were 3 tall and 1 dwarf. Tall and dwarf plants were in the ratio of 3:1. This ratio of 3:1 is called monohybrid ratio. So just imagine, the first generation had all the tall plants, but the second generation has 3:1 ratio of the dwarf plants. So where were these dwarfness gene in the first generation? It could not express itself. So that is what Mendel suggested, that the dwarfness is a recessive gene which could not express itself in the F1, but it was able to express itself in the F2 generation because there were no dominant gene there. So that is this gave rise to first law of Mendel. Mendel suggested that during gamete formation, the chromosomes, the genes, they separate out, they segregate to form half-half to each cell. This is called law of segregation. During gamete formation, the genes, they segregate and only half of the gene enter into each gamete. And during sexual reproduction, two different genes combine together to form a new organism. And this ratio of 3:1 in second generation F2 progeny is called monohybrid ratio. Now let us move on to the next type of experiment, which is dihybrid ratio. Now, the second type of experiment performed by Mendel was dihybrid cross. We just discussed about monohybrid cross. This is dihybrid cross. In this, what happens? Two plants, there is cross breeding between them, considering two pairs of contrasting characters, right? So in monohybrid, we considered only one contrasting character, like tallness and dwarfness. In this, we'll take into consideration two characters. So during this crossbreeding, what was noticed, that came out as a law of independent assortment. During gamete formations, the genes, they separate out, that is law of segregation. Now, one pair of genes for one character, they pair with another pair of genes of another character independently. That is second law of Mendel, law of independent assortment. Let us study how it happens. Now, for example, I have considered an example of round green seeds and another parent having wrinkled yellow seeds. Now, the seeds are either round or wrinkled. This is one character. Since round is a dominant gene, I will take capital R as a symbol. While wrinkled is a recessive gene, we'll take as small r as a symbol. On the other side, another character is the color of the seeds, which is either green or yellow. Yellow is a dominant gene, so I have taken capital Y as the symbol, while green color is a recessive gene, so small y for this. So this plant has capital R and small y, while the other plant has small r and capital Y. Now, during gamete formation, the same types of gametes will be formed, one from R and another small y. So this gamete will have one gene for round and another for green color of the seed. While in this case, the gametes will have small r for the wrinkled character and for yellow, capital Y. So these are the gametes formed. Now during cross breeding between these two plants, what would happen? If this gamete fuses with this, what will you observe? Capital R and small r from here. Then capital Y and one small y from here. So, since there is one capital R, so the seed will be round. And one capital Y, the seed will be yellow. Right? Similarly, if this gamete fuses with the other gamete, if there is a chance fusion, it will again be R, R, Y and small y. Then this between these two gamete, again same thing. These two gametes. So you'll notice in F1 progeny or the first generation, all the seeds are round and yellow. So that is why we considered round as the dominant gene. Similarly, yellow color as the dominant gene. While the wrinkled, the small r, is a recessive gene. It could not express itself in the first generation. Similarly, small y is for the green color of the seed. Even that could not express itself in the first generation. So all the plants bear seeds which are round and yellow in color. This is the F1 progeny. Now, these plants were allowed to have self-pollination. What types of genes will be formed? This R can pair with the capital Y, capital R with capital Y, right? This is the first type of gamete. Or capital R with small y. This is the second type of gamete formed from the F1. Then the small r may pair with the capital Y. This is the third type of the gamete form. And lastly, small r with small y. So these are the 4 different types of gametes which were formed from the F1 generation. During self-pollination, either of these will fuse together to form the F2 generation or the second generation. Now, just try to do it yourself. If this fuses with this gamete, what will you observe? Capital R, small r. So it will be round. Capital Y, it will be yellow. So 4 types of, we'll just discuss how 4 types of the plants were observed. Although we took only 2 types of parents, but in the F2 generation, Mendel found 4 types of plants. First, round and yellow, then wrinkled yellow, then round green and wrinkled green. So 4 types of seeds were observed in the F2 generation. The ratio was 9:3:3:1 of these 4 plants. This ratio is called dihybrid ratio. 9 out of 16 plants, this total is 16. So 9 out of these 16 plants were round and yellow, that is both the dominant genes. 3 were round and green, 3 were wrinkled yellow, but only 1 was wrinkled and green in color, both the recessive gene. This 1 represents wrinkled green, both the recessive genes. Now, in the tabular form, I'll explain you how we found this ratio. Now, consider these 4 as the gametes of one parent and these as the gametes of the other parent, the male gamete and female gamete. Now, if this capital R, capital Y fuses with capital R, capital Y, we'll find this F2 progeny. This will be round and yellow. Again, this fuses with this gamete, capital R, capital Y, round yellow. So this way you can frame the 16 types of plants formed in the F2 generation. Out of these 16, you'll notice 9 of them have round yellow seeds. If there is even one single capital R, the seed will be round. Even if there is one capital Y, the seed will be yellow, right? Because they are dominant gene. They'll express themselves even in the presence of recessive gene. Now, this is the first type of plant, that is round and yellow. We found them to be 9 out of 16. The other type would be round and green. Let's see. This one is round and green. This one is again round and green. One capital R, so round, but both are recessive, so round and green. And the third type, if you can find out, round and green. This round for capital R, small y will represent green. So 3 of them are round and green. Then comes wrinkled and yellow. Okay, this one, wrinkled and yellow. Small r will represent wrinkled, capital Y will represent yellow. So wrinkled yellow, and this one again wrinkled yellow. So 3 of them will be wrinkled yellow. But wrinkled and green, both recessive, you will find there is only one, this one, wrinkled and green. So the ratio is 9:3:3:1. 4 types of plant in the ratio 9:3:3:1. This is called dihybrid ratio. From this, Mendel could tell us that during gamete formation and during fertilization, what happens? The pairs of the genes, they separate out and they form new pairs independently. Okay, so that is law of independent assortment. Now let us move on to the next topic. Students, the next topic under heredity is sex determination in humans. Now, whether the child will be a girl child or a boy child, this sex determination in human beings is genetically controlled. As you know, we have 23 pairs of chromosomes. Out of these 23 pairs, 22 pairs of chromosomes are called autosomes. They control the normal functioning of the body, like the characteristics, the external features, or the other systems, digestive system, respiratory system. They are controlled by the other 22 pairs. But there is one pair of chromosomes which are called the sex chromosomes. They play a role in determining the sex of the newborn baby, the new offspring. Now, in males and the females, that is both the parents, 22 pairs of chromosomes are identical, they are similar. But the 23rd pair is called the sex chromosome. It is different for males and different for females. In males, it is termed as XY chromosomes, but in females, both the chromosomes are identical, so there is X and X. So during gamete formation, they separate out, the pairs separate out to half the number. So in males, the gametes have, half of the gametes have X chromosome, while the other half have Y chromosome. But in female, when gametes are formed, the gametes are similar. All the gametes will have the X chromosome. When you make it into half, X and X. So all the egg cells, that is the female gamete, will have X chromosome, but the sperm cells, half the sperm cells will have X chromosome, the other half will have Y chromosomes. Now what happens during fertilization? One of these sperm cell will fuse with the egg cell. So if the X chromosome of the male fuses with this female gamete, the next zygote will have XX chromosome. This zygote will grow up to a girl child because it has XX chromosome. But if the sperm bearing Y chromosome fuses with the egg cell, it will have X and Y zygote. This zygote will grow up to be a boy child, right? So this is how the sex is determined for the new offspring in human beings. So that means whether you will be a boy or a girl, whether the new offspring will be a boy or a girl, it depends upon the genes which it has inherited from the father, from the male parent, not from the female. So it depends upon the sperm which fuses with the male gamete, with the female gamete. That is how the sex of a newborn baby is determined in a human being. I'll take another example of blood group. The blood group is controlled by three genes which are represented by the symbol IA, IB and IO. It is represented like this. So, out of these three, A and B, they are dominant gene, while this O1 is a recessive gene. So as you know, there are four blood groups, A, B, AB and O. So if a person has a blood group of A, it might be because of presence of this gene, right? Both are dominant, or it might be because of presence of a pair of gene like this. Since O is recessive, if a child got O from the father and A from the mother, it will have A blood group because O is recessive and it will not be able to express itself. But this A gene is dominant, so it will be able to express itself. Similarly, if a child has blood group B, that can be because of both the dominant genes, or it can be because of one dominant B and the other recessive O gene. If a child has blood group AB, this is because both the dominant genes are present, A as well as B. Both are able to express itself. I told you, both are dominant gene. So the child will have the blood group AB. But to have a blood group O, both the genes need to be recessive. Then only the child will have the blood group O. If this is present with a dominant gene, then it will not be able to express itself. Recessive genes can express themselves only if both the genes are similar. That is why only this child will have a blood group O. So, if you have a blood group A, that doesn't mean that you have both the dominant gene or both the parents need to have A. It can come from this combination also, right?